Thanks so much. Thanks for coming, and thanks for inviting me to this exciting workshop. It's really cool to be here. I'll jump right in. So for this project, we started with a really simple question, namely, how do intelligent agents know what's good for them? And when we say intelligent agents, obviously we mean ourselves, right? So how do we know what's good for us? And how do we know which actions to repeat because they were beneficial in the past? And how do we know when the screen is perfect to continue with your talk? So obviously the standard answer to this, if I can manage the technicalities. Yay. The standard answer to this is obviously the reinforcement learning framework, right? So we think about intelligent agents as being embedded in the environment and they're interacting with an environment. They're emitting actions that potentially change the state of the environment, and then they're receiving information from the environment about these states, but also separately, this other signal that we call the reward signal, which is basically a thumbs up or a thumbs down from the environment, right? And these signals the agent can then use to learn, which actions tend to lead to rewards, which states in the environment are associated with high rewards, and then the agent can change its behavior in clever ways to maximize reward, or at least some function of it. Now, of course, it's been pointed out that if we apply this image as it is to biological agents, there are some weird things. It's slightly paradoxical, and there's multiple issues, but the most prominent one is, why is the reward coming from the environment, right? How does the environment know what's good for the agent? The reward is subjective, obviously. What is rewarding to me will be very different from what's rewarding to you, and it will change over time, depending on where I am, my current state, but also my goals, my desires, my needs. Yeah. So the reward function is not constant in biological agents, and it cannot be generated from the environment. It needs to be generated within the agent. Okay. And of course, this is actually not how we were ever supposed to apply this framework to biological agents. If you instead look at this image on the right, you can see that if we think about organisms, then actually this whole thing that's in gray here is supposed to be the organism. So the RL agent that we're talking about is but a submodule of the organism. Yeah? It's that part of the brain that does the learning from rewards. It's not the whole brain, and it's not the whole agent. There's other things that are going on in this organism or in this brain, such as this part here, which is actually generating the reward signals. Yeah? So the correct picture is that there are sensations coming from the external environment. They provide some information, but this information does not have valence per se. The valence needs to be generated within the organism, right? And I think there has been some confusion when we only look at the image on the left and apply that to experiments with humans or animals, because we kind of assume that we're designing the reward, right? We're telling the participant, oh, you earn 5 points now for this action. And then we think we know what the reward is for the participant. And then when they behave, and they behave differently than other people, we interpret these differences in terms of learning from rewards, learning rates, or reward sensitivities, and those kinds of parameters, when actually we don't actually know whether it felt like the same reward for them. And so that's one side of the coin. It's a bit confusing, but there's something more subtle. It seems that we've made this conscious decision to say these are two separate modules. There is one part in the organism that is responsible for generating the reward signal. And then there's this completely independent part, seemingly, that then takes that reward signal and learns from it, which assumes that it does work independently, which I don't think has been tested at all. But also, it seems to suggest that the locus of intelligence is the reward learning, right? There's this implicit assumption that if you think about primary rewards, for example, the transformation of external sensory stimuli into valence signals is probably working via some hardwired pathways. Yeah, we taste some sweet sugar or something, and then immediately there's a reward signal. And that pathway is not particularly interesting for intelligence. What is interesting is how we then learn from the rewards, how we change our actions and build models of the future, etc. However, I would argue that we're missing out on quite a big, really important part of what biological intelligence is. Because if you only look at the RL agent, there's no goals, right? If the reward function is designed by the engineer, it means that the agent will maximize reward, but the goal that it fulfills by doing so is defined by the engineer. And what makes biological intelligence different is that we have control over our reward function. So I do think it's quite a critical component. And so Debbie and Rike and I were kind of wondering about this and starting to wonder, what do we actually know about how primary reward signals are being generated in brains? Do we know anything about the circuits? And is there anything interesting? So we started to talk to Dana Small, who's an expert in the neuroscience of ingestive behavior. And we went down this rabbit hole of ingestive behavior, which I had no idea about previously. It was super exciting. And I'm just going to share with you very briefly and very superficially the things that we learned from this journey. So first of all, it seems that we have this classic view of conditioning where we think, if you consume an apple and you get the sweet taste in your mouth, that's where the reward is, right? It's kind of linked to the consumption of the item. And so immediately, you lick your ice cream and wow, that's a great experience. So that's going to reinforce buying the same ice cream at the same store next time. Okay. However, and we also know that actually there is activation, phasic activation of midbrain dopamine upon the receipt, upon the sweet taste or whatever taste you like in your mouth, right? So there is a corresponding physiological signal, and we know the properties of these. We know it travels back in time over learning, so that you are then the reward prediction error happens in response to the cues that predict that unconditioned stimulus. So that's the classic view. However, there's been some seminal work over the last decade in beautiful animal experiments showing that it's actually not that simple. This is not how it works. So for example, if you give mice an artificial sweetener, so they do get the sweet taste, but they don't get any nutrients, then the behavior, the licking, but also the dopamine response will not be sustained over time. It will stop at some point. Yeah? So the organism will learn that, yeah, it does taste sweet, but there's no benefit that I'm having from drinking this solution. And even more interestingly, you can look at sweet taste blind mice. So these are mice that lack the cellular machinery required for sweet taste transduction. So they will not prefer a sweet tasting solution over water at all, as compared to wild type animals. But they are capable of conditioning a preference for a flavor based on the sugar content of that solution. So they are able to learn via some other mechanism. But it just goes to show that sweetness is neither necessary nor sufficient for reinforcement of behavior. So then you can ask, what is the mechanism? What is necessary and sufficient? And it turns out that for sugar, it seems to be glucose metabolism. It seems to be actually the conversion of sugar into energy that is sensed in some way by the body, and that actually elicits the primary reinforcing signal for sugar. So here you see that when we give animals sucrose, for example, we've got an increase in striatal dopamine under normal conditions, but if we impair ATP production, so the animal still receives the sugar, but it cannot convert it into energy anymore, then that dopamine response goes away, as does the reinforced behavior. And there's several control conditions for this that show this, but it basically shows that it is actually really the conversion of sugar into energy, which is something profound about what is good about that resource, right? It will be rewarding to the extent that we can use it for energy. And actually, there's a sensor in the hepatoportal vein, so that's just a blood vessel that transports the nutrients to the liver, and it actually detects this metabolizing of sugar. And then that signal is being transported via the vagus nerve and ends up in the striatal dopamine release. So we've got all these elements here. We've got the resource itself, so the carbohydrates. We've got some peripheral sensing mechanism, and a signal that then leads to dopaminergic activation as well as reinforcement of behavior. And then we looked at other resources. So this is the sugar pathway, but you can actually draw very similar pictures for fat rewards and also for water. There's always a peripheral sensing mechanism that is different across those resources. So for fat, it's the detection of the nutrient in the upper intestine. And for water, interestingly, similar to sugar, it's a very downstream consequence of consuming water, namely rehydration. Yeah, it's systemic blood osmolality that is being detected, and that actually generates the final reinforcing signal, the necessary condition for reinforcement. So across these resources, we've identified these pathways in animals that clearly map out how the resource is being detected, how it's evaluated, and then transformed into a reward signal. And I'd like to show this video for the example of fat rewards. So what you see here is a mouse, and it's poking its nose into these openings. And whenever it pokes the nose into the opening, then it doesn't receive anything, but the experimenter will optically activate those neurons that detect the nutrient in the upper intestine. So those vagal sensory neurons that innervate the gut and that are responsible for the detection of fat in the gut, right? And you can see that this stimulation, this optical activation of this peripheral pathway is sufficient to induce self-stimulation. And they've also shown that it's sufficient to induce place preferences and flavor preferences and all those hallmarks of a reward neuron, basically, right? But remember, it's not a neuron in the brain. It's a peripheral sensor that is responsible for detecting fat. And what's also interesting about these different pathways for the resources is that they are quite parallel, but they're still segregated. So it's different neural populations all the way up that code for the detection of sugar and the detection of fat, for example. And even behaviorally, we can see that they contribute to reinforcement individually or independently. So mice and humans will prefer a solution that comes with both sugar and fat, even if that mixture in the solution is matched in terms of the total calorie content to a solution that only has sugar, only has fat, showing that it's really not a property that's inherent to the stimulus that we're consuming, namely the calorie content, but the driver for reinforcement is the internal signal produced by the body. And because it's producing two separate signals, it will be more reinforcing if it has both. Okay, and the similar story for water, but I'll cut it short here. Basically, what we need to do is we need to update this view of what's the unconditioned stimulus, because clearly the oral signals are not. Yeah, but this updated view raises some really interesting questions, and we focused on 3 of them in our project, and I'm just going to briefly share them with you. So first of all, if the oral signals are not the unconditioned stimulus, what are they? What function do they serve? They're also not a conditioned stimulus, which we'll talk about in a second, but what do they do? The other question is, how do they interact with the late signals? There has to be some, very careful coordination between those two, because, the animal will be successful if they correspond to one another in most of the cases. And then finally, we said in the beginning that we actually want a reward function that's dependent on the internal state and the goal of the organism. So if we look at these newly discovered primary reward signals, how they're impacted by the internal state of the animal. So to look at the first question, we thought a little bit about what these oral signals could be. And they are privileged to some degree. So sweet taste is not a conditioned stimulus. It's innate. Yeah? We get these reactions to sweet taste in newborns of many species. So there is something hardwired about them. But they're also not the unconditioned stimulus, because if we decouple them from the post-oral reward consequence, then their reinforcing strength will decrease. They will not be reinforcing anymore. So they are something intermediate, and they're also physiologically different from the late primary reward signals. There's also something really intriguing about those signals that have been discovered, is that they tend to end up in the dorsal striatum, whereas the early reward signals elicited by the oral signals, they tend to end up in the ventral striatum. So it's a separate circuit and presumably has a separate function. There's been a really interesting proposal by Peter Dayan a couple of years ago as to the function of these early signals. And the proposal goes, if you think about it, you consume the food, and then only tens of minutes later is your body actually able to evaluate the ultimate consequences of that action. The ultimate reward, after digestion, when you detect the systemic change in your body, is quite delayed with respect to the action itself. And that delay poses an interesting credit assignment problem, because many things could happen in between the action and the outcome, right? So you need to make sure that the animal is able to learn to associate that late outcome with the action of consuming. And that's where these early signals are extremely useful. So you can think of them as shaping signals, as we do in other kind of complex situations where the agent has to follow multiple steps until in the end there's a sparse and rare reward signal. The shaping will help immensely. So it's like a little nudging signal on the way that you're on the right way, and it helps to improve the speed of learning massively. So that's a potential computational function of these. Now, when it comes to the interactions, there's multiple interesting things to say. I already said that the licking and the dopamine response to the early oral signals is not sustained. So there has to be some feedback mechanism. And we've been knowing this for a long time. Like Holman did some experiments in 1969 where he decoupled the post-oral consequences of consuming a solution with kind of the flavor and the oral signals, and already showed that this is updating the early response. But interestingly, the arrow goes the other way too, because if we taste something sweet, this will also elicit a metabolic response that will prepare the organism optimally for the predicted receipt of the nutrient in the stomach, right? And that even happens for earlier signals. So upon the sight and the smell of food, there's already predictive mechanisms, prospective control mechanisms, such as insulin release or the suppression of AGRP neurons, which actually change metabolism. And this is actually a bit mind-blowing because it means that the primary reward signal that you get in the end will be different depending on how well you were able to predict it, right? So lots of interesting open questions as to these interactions. I'll say one last thing about how the internal state shapes those reward signals. And we don't know a lot about this yet. We know a lot about that internal state changes these early signals because we've been measuring them forever. We know they're different when you're hungry than when you're satiated. We also know that there's interesting differences in how we process food cues depending on the internal state of the animal. But what we were really interested in is how do these primary reward signals get generated depending on the state of the animal. And we identified kind of two primary mechanisms or patterns here. The first is relatively intuitive. We call it a state-driven reward signal. And the way you can think about it is if you think about thirst or drinking, you have this homeostatic state that you want to be in, which is you want to be hydrated, right? So you have a goal or a homeostatic set point. And your current state might be somewhat away from that. So there will be a distance between where you want to be and where you currently are. Then you take an action, and that action might actually get you closer to that goal. So the distance between you and your goal in that toy one-dimensional space here will be reduced. And that reduction might actually just be the source of the reward signal. So the action might be reinforced to the extent that it brings you closer to your goal. And that's kind of a drive reduction or distance to goal reduction mechanism that would be responsible for generating the reward signal. But there's also a different mechanism that seems to be relatively independent of the internal state of the animal, and we call that an appetition event. And that's what we've seen for sugar and fat, where there's really a specific physiological event that does not correspond to a state change in the animal, but it still is able to elicit a reward. Now, the first of these, the state-driven reward signals, they are predicted by a framework that's called homeostatic reinforcement learning. So that's basically the formalization of this drive reduction idea, this distance to goal reduction in high-dimensional spaces. So you just have to find where you are with respect to where you want to be. And then you can measure kind of how much closer does the action bring you. And then you can quantify that distance and you can use that signal as your reward signal. And actually that's exactly how it works, right? You define a landscape where you are and where you want to be. You measure how much further any action brought you to your goal. And then that's what you plug in to your usual RL, right? So again, this reflects this idea that these processes are completely separate, right? You can have some reward generation mechanism, and homeostatic RL is really beautiful in laying that out explicitly. But then the assumption is that signal is just going to be fed into your RL agent, just as we used other reward signals previously. I'm not sure that, we know that that's the case. But anyway, that's the suggestion. There's other things that this is missing out on. In particular, as I said before, if we look at the mechanisms for sugar and fat here, for water, you can really think of this as a drive reduction or a change in the state. But for sugar and fat, that's not the case. And indeed, sugar remains reinforcing beyond satiation. And if you think about it, it makes a lot of sense because we can store sugar and fat, right? So it would make sense to still reinforce actions that acquire energy, even if you currently don't need it. That's different for water. You cannot store water. So water really should only be rewarding when you currently need it. And that is a logic that whether you can store a resource or not, that might actually generalize to more abstract rewards as well, and is an interesting dimension to think about. Okay, so putting it all together, this is a slightly convoluted picture of how we could redraw the reinforcement learning framework that I showed you in the beginning. So we still have this agent here in the center, but this agent now in our view corresponds to the whole brain, right? So it's not just the RL agent, it is actually the whole, intelligent bit of the organism. And it has an RL mechanism in there for sure, but it also has these sophisticated ways of actually computing the R, computing the reward signal. And there's a lot of work that we can now do to quantify how does that depend on our priors, on our state inference, right? Because I said for some of these rewards, the reward signal is actually a function of an internal state change. So the brain actually needs to track what state the body is in, and it needs to be good at it, right? It might make mistakes, and then you could have a reward signal that is actually more of an illusion than a real benefit for the body. So a lot of interesting questions here. And then obviously this brings to the front again the mechanisms of inference that I'm really interested in. So I think marrying this idea of state inference with the idea of affect and valence and reward will be really critical in the future. I also hope that, we've looked at primary reward signals, but I think we can draw some conclusions more generally for circuits implementing goal-dependent abstract rewards. And there's a couple of interesting things to take away. So we have this issue of timescales, which is, for example, not included in homeostatic RL at all. There is this mechanism of having a shaping signal early on and then bridging across the delay to the final reward signal. We've also seen that it seems to be more useful to think about modular rewards that kind of, almost reinforce behavior independently rather than this final scalar common currency idea of a reward. And then this idea of storage has emerged as maybe one fundamental dimension that we need to think about. And so I would propose that if we try to design intelligent agents, that we take this into account and move away from just feeding the reward to the agent, but thinking about how an intelligent agent would produce intelligent reward functions and reward signals. And these are some of the things that I'm working on at my new lab in Osnabrück, which is a small German city, not very famous, but it has a great Institute for Cognitive Science. So here I'm heading this Models and Interventions for Cognition lab. I'm also part of this new initiative, the MIND Initiative for Machine Intelligence and Neuroscience Discovery, also at Osnabrück. If you're interested to contribute to any of these, do reach out because I'm currently building my lab there. And with that, thanks so much for my brilliant collaborators who are also here partly and for the audience. Thanks.
Yeah.
Thanks. That was really interesting. Thanks. That was really interesting. I was wondering, in this new era of multi-modality and getting as much data as possible, of course, it's a systems neuroscience conference, so we like neural data. But we saw, for example, how important other organs are in the body. So would you advocate for collecting data through other sensors in these cognitive tasks? And where should we focus on beyond just neural data? For example, could we have glucose monitoring? Would that be an important thing in your opinion?
Yeah, that's an excellent question. I absolutely would promote for that. However, I think it really depends also on the kind of reward that you're interested in. My suspicion is that there will be interesting influences between the bodily state of the organism and the way that you even process abstract rewards, such as points in a game, because I do feel like the circuits that will process these abstract rewards will build upon, the older circuits, and so dimensions such as storage will actually still be relevant, and I'm sure there will be interesting interactions. But if you're only interested in the abstract reward and not, and you don't have a strong hypothesis for how that interacts, you might not need the additional measurements. But if you're interested in primary rewards, I would say absolutely, yes.
We have time for one more question.
Thank you very much. Super interesting talk. So I was wondering why, in the end, you didn't decide to go all the way through and just integrate the body within the environment to do to think about it in this particular way. Do you feel like there's something that makes it significantly different that we should consider it as a third part of RL and not just included in the environment?
Yeah, that's again an interesting question. So in this image, we've put the body kind of in between the agent and the environment, right? And I do think it is a privileged part of the environment. I don't think you can equate it with the external environment, because if we think about what the primary goal of the brain is, evolutionary speaking, it's to keep us alive, right? And so whatever happens in our body will be relevant and much more relevant than what happens outside the body. So the brain is still responsible for all those variables that are being tracked in the body, whereas the external environment is only relevant in the sense that it interacts with our body, right? So I guess it might not make a difference for the computations, depending on how you write it up. But I think it makes sense to think of for us to think of it as a privileged part of the environment for sure.
That's a keyword.
Thank you.